So everybody, thank you for attending our last webinar session of this year. Today I have the pleasure to host Dr. Maria Corradini from University of Amherst, Massachusetts. Dr. Corradini is well known for her modeling efforts in predictive microbiology and has been working in recent years with digestion and measurement of optical properties in foods. So Dr. Corradini, thank you for being with us, and you may start when you want to. Thanks.
Thanks so much, Vinicius, for your presentation. Good morning, afternoon, or evening, depending on your time zone, to all of you. And thank you for joining me in this webinar. During this presentation, I will introduce a diverse set of optical approaches that can contribute to research and development, as well as quality control in food science and engineering. These optical approaches mostly revolve around the use of luminescence spectroscopy and fluorescence microscopy, and the examples that I'm presenting correspond to applications developed by me and my collaborators. Let's start first by making a case for luminescence spectroscopy in food applications. What is luminescence spectroscopy? So, one of the main advantages of luminescence spectroscopy is that it can report at the molecular level. So basically, it reports on the molecular environment. When we are considering analytical techniques, we have been extremely efficient at developing strategies that permit us to access the macrostructure and bioproperties of foods. Also, developing strategies that allow us to figure out what happens at the micro and nanostructural level. However, when it comes to molecular structures, it's quite difficult to obtain information, and usually, quoting Dr. Campanella from Purdue University, probably our best option would be to modeling. However, in the case of luminescence spectroscopy, you will be able to infer and have some data at the molecular level, and also infer how the X of the molecular environment affects either the micro and macro structures. Only luminescence spectroscopy proposes some advantages in terms of reporting at the molecular level. Also, it's a non-invasive technique that can tackle the sample as is. Of course, there are certain caveats that have to do with sample clarity, but those can be circumvented using specific modes. Also, due to the versatility, availability, and handheld devices, and the time required to perform tests in most cases, most of the measurements can be performed in real time, and also provide spatial resolution. Means that it will be able to out what happens in a heterogeneous sample and provide information from different parts of that particular sample. However, probably one of the most interesting parts of this is that there are a huge amount of food-grade fluorophores that are already embedded in foods. So basically, they can be used as sensors and reporters of microstructure, component interactions, quality and safety within the food. You ask me how spread these fluorophores and in food. Let's take the case of a simple plate of salad. Mixed greens, curry chicken, cucumbers, cheese, pickles, and golden beets. Once exposed it to a full spectrum light, you see, of course, what we normally see when we eat our food. But once we excite them with UV light or UV light, you see that our sample actually glows. So our foods glow. You can see the emission from the curcumin in the curry chicken, the chlorophyll in the pickles, the riboflavin in the cheese, and also the beta-xanthine in the golden beet. So one example is quite simple, such as, for example, a solution of quinine or a solution of riboflavin, or complex ones. They can emit a particular glow. In this case, for example, in the case of red lentils from Monsanto fires, and lutein in the case of yellow beets from beta-xanthines, and in the case of candidators from luminescent color such as fast green. So you can see what happens when we expose them to blue light or UV light. You can see a persistent glow that spreads in most of the samples. So, but let's talk a little bit about photoluminescence. When we are talking about luminescence, we are referring to the emission of light by a substance not resulting by heat. Most of you probably already know what are the differences between fluorescence and phosphorescence, but just let's refresh this concept before continuing this presentation. When we are talking about photoluminescence, we are talking about a compound, and we are going to describe it in terms of the state energy diagram. So we are going to have a compound that, or a particular molecule that is at a ground state. Of the molecules that exhibit luminescence usually are aromatic compounds or are composed of conjugated dienes. That sample is excited by the addition of light. It moves to a higher energy level, and that excited singlet state is characteristic of that transition. Of course, as molecules, as ourselves, we cannot be excited forever. Therefore, we have to go back to ground state. A case of molecules that can be performed by two pathways. One that involves no emission, which is always or most of the time related to motion, and another one that it implies the emission of a photon with radiative pathway that results in fluorescence. Alternatively, the excited state can transition a process called intersystem crossing to another high energy state, which is a triplet excited state, from which, of course, it has to decay in order to reach ground state. The triplet excited state with by motion, of course, is non-radiative. What happens with the emission of an photon is called phosphorescence. So basically, we have two processes that are exploiting in these optical techniques that are fluorescence and phosphorescent emission. The alternative decay, radiative pathway or radiative pathway, is what determines the environmental sensitivity of this probe. In terms of photoluminescence, there are certain photophysical properties that characterize it. Let's review those before continuing. One relates to the intensity of the emission and the quantum yield, which correlates to the probability that photons are emitted. Although this one can be altered by the structure of the compound that we are using, for example, in this case, if we have pyrene, the addition of butyl groups to pyrene increases the quantum yield of the sample. However, it's mostly affected by quenching reactions between the excited molecule and the environment. Remind you that the quantum yield corresponds to the ratio between the number of photons emitting and the number of photons absorbed by a particular compound. Among other important properties are energy, which basically represents the wavelength distribution, or the core of the emitting photon. As you can see in this figure, the environment might affect the location of the peak, and consequently, of course, the energy that emission carries. This is particularly modulated by bonding interactions between the excited molecule and the environment. The lifetime, which is the time delay between absorption and emission, is one of the most important characteristics that we're going to discuss in this presentation, and it can be used by quenching reactions between the excited molecules and the environment. So basically we know that the particular optical properties, intensity, energy, and lifetime are being affected by environment per se. Make these probes sensitive to what happens around them. So when we are talking about photoluminescence, probably you are familiar about the numerous uses that it has been in chemistry and biology. For example, the use of solvatochromic dyes to characterize solvents, the use of green fluorescent proteins, and also there are applications of fluorescent dyes, selective fluorescent dyes in microscopy that allows to differentiate, for example, a lipid region from a proteic region in a cell. However, when it comes to foods, use of luminescence spectroscopy basically was devoted to composition, authenticity, and, for example, the identification of contaminants, as, for example, in the case of mycotoxins in corn. The idea is to expand applications in food science and engineering. So in order to do so, this presentation will focus on 4 aspects: the development of sensors of food quality, particularly with the use of molecular rotors, which are characteristics and sensitive to molecular crowding, which allows them to be sensitive to the local and bulk viscosity in biophysics of digestion and fat crystal networks. I also will devote some time to other applications we haven't performed by my group, however, that might be interest for some of you, such as fluorescence lifetime imaging or FLIM, the use of molecular rotors in flow sensors. Thus, I'm going to discuss diffusion using fluorescence discovery after photobleaching or FRAP, and also the simultaneous diffusion of luminescent probes and specific components using FRAN cells. I will devote most of the time to characterization or under characterization, in particularly the case of delivery systems. And I will introduce the concept of phosphor or fluorescence resonance energy transfer, or FRET. At the presentation, I will make remarks in terms of the use of luminescence spectroscopy in shelf life monitoring using hyperspectral analysis. I'm not going to devote too much time to these issues since there have been extensive reviews by Sung or Valgramidis in the last 2 years that deal with this subject. I'm going to devote some time to molecular rotors, SLAP and FREP, funny as the name sounds. A little bit with the use of luminescent probes as sensor for food quality and probably food safety. So the hypothesis is quite simple. We already established that there are a large amount of fluorophores in foods, and that we also add additional fluorophores to them, for example, in the case of additives. If we map their environmental sensitivity by linking their changes in their photophysical properties with particular factors such as, for example, pH or water activity, they are no longer random components in food, but they can become now sensors of food quality or food safety. This is based on that the chemical composition of the surrounding medium has in the fluorophore that we are recording the emission. Sometimes that we excite this particular fluorophore, the composition affects emission, so now it reflects, if, for example, of pH or concentration of sodium ions in a particular sample. So see that this particular fluorophore is sensitive to the pH of the environment, so it is to a pH 9, its emission intensity is much higher than when it is exposed to a pH 5. Of sodium, the presence of sodium ions, this fluorophore also is sensitive to its concentration. So basically, the photophysical properties provide molecular information about the food. They do it with rapid response time, with no alteration of the sample during testing, while also in line testing. That physical composition affects the emission, but although the physical properties of the surrounding medium affects it as well. So basically, we can say that during the last few years, I have devoted time to sort molecules with lines and them screen. However, I didn't limit my research to that. Basically, what I try to do is translate the screen into useful information. In the last 3 years, we have identified around 15 particular probes or lucent probes that has diverse sensitivity to characteristics of the environment. Most of them, as you can see here, provide information on the physical state of food, such as rheological properties, as for example, you can see for most of these probes. And also other characteristics such as water activity and hydrogen bonding ability. The ability of these molecules to be sensitive to the physical state because most of them exhibit rotor behavior. Let's talk a little bit about molecular rotors. In molecular rotors, we are talking about molecules that have usually two segments to rotate around each other. This determines that upon photoexcitation, they have the ability to exhibit two states, a planar state and a twisted state. That makes it sensitive to changes in free volume, and consequently, they make them potential probes of physical states of foods, reporting on microviscosity, aggregation, microstructural organization, interface fluidity, and polymerization. So, if we see the proposed mechanism for the molecular rotors, we say that upon photoexcitation, there are two states, as I mentioned before. One local excited and planar singlet state, and a twisted state. The environment around the probe is quite liquid or fluid. A transfer from the locally excited state to the twisted state is quite easy, and rate of formation of this twisted state is quite high. However, if we impose any kind of restriction in movement, this transformation to the twisted state is hindered, and that makes the local state the most prevalent, which usually decay in a radiative form, exhibiting an increased fluorescence. It mentioned also that in the case of the twisted state, usually or non-radiative way when it goes back to the ground state. So, if we are in a fluid environment, the state is the prevalent, and consequently we have radiation relaxation. However, any movement in restriction results in the prevalence of locally excited state and consequently an increase in fluorescence. We want the first ones to observe this kind of behavior. molecular rotor has been commonly used in biology. For example, BCBJ and transestilbene has been reported in order to assess fluidity and characteristics of the interface in cells. However, of these molecular rotors, which are basically synthetic, exhibit stability, limited stability, and potential toxicity. So one of the ideas is to use glass or edible probes to have this behavior, but we are going to have increased stability. They are going to be soluble in large range of systems because, of course, we have glass probes that are liposoluble and also hydrosoluble depending on the food that they have been applied. Of course, they have increased safety because they have been tested for human consumption. Not only will affect cytotoxicity when applying this kind of techniques in cells per se, but also allow them to be always present in foods and be consumed because that's what we are normally doing, and can be used in order to monitor statistics in real time. Molecular rotors that we have identified as glass probes are many of the approved synthetic food colors currently in foods: high soluble azo-dyes, insoluble azo-dyes, and triaryl methane. Of course, in order to test the capability of them to operate as molecular rotors, they have to withstand the typical experiments that are being performed in order to test a synthesized probe as such. So basically, these kind of experiments involve in the sensitivity to the viscosity of the medium in terms of their fluorescent intensity. So basically what you do is you include that molecular rotor into a solution of different viscosities, measure the fluorescence intensity of them. This is a conventional molecular rotor that is normally used in research, and these ones are two synthetic dyes that we thought that they have presumptive molecular rotor behavior. And in all the cases, we observe an increase in intensity as the viscosity increase, not only the micro viscosity but the bulk viscosity. This has been evaluated in glycerol water systems, which is normally what is being done for molecular rotors. Also, the relationship between normalized intensity and viscosity being characterized using the Forster-Hoffman equation, and relationships for the synthetic dyes similar to the one that is obtained by one of the conventional molecular rotors. These molecular rotors are evaluated on value of the alpha parameter, a viscosity sensitive meter. There are the values that we obtain for these three rotors. You can see that in the case on the edible ones, the results are lower, so show less sensitivity towards viscosity. It's not markedly different, and usually is considered an acceptable sensitivity is observed when you have values of the alpha parameter above 0.2. However, we need to expand the definition of these edible probes as truly molecular rotors, because we will have to differentiate whether they were only sensitive to microviscosity or if there were other factors that were affecting them, the polarity of the system. So when we see viscosity, this one corresponds to the excitation spectra Allura red, a synthetic food dye in ethylene glycol and glycerol. Glycol at the same temperature has, of course, a much higher viscosity, and of course, the emission is much higher for it. So we wanted to test what happens also when we change the polarity of the system. And we that all they have high sensitivity to microviscosity, they have moderate sensitivity or low sensitivity to the medium polarity. Allows us to say that in principle, these fluorophores are sensitive to microviscosity. However, there is a minor problem. Using just fluorescence emission intensity measurements to sustain this claim is not very reliable because the intensity or fluorescence intensity is concentration dependent. We have to have another kind of measurement that allows us to figure out if the concentration was not what was determining that increase in fluorescence intensity. So we use lifetime. It is much more difficult to conduct at fluorescence intensity, and that's why it's not regularly conducted, especially for food applications, especially because the requirements of the equipment are also much more complex. In water, you see this one is the impulse response factor, and the impulse response function. And this one in blue is one in red is density of Allura red in water. So basically, we couldn't see much difference. But when we change the Allura red and we put in glycerol, we see an increase in lifetime of this particular molecular rotor. We tested not only with Allura red, but with the whole set of synthetic and natural dyes and cases. We obtained that by changing the viscosity with the measurements of the lifetime. So, as viscosity increase, the lifetime of the synthetic dyes that we are mentioning in this particular study increase significantly from, for example, in the case of Allura, 2 picoseconds to 200 picoseconds, and in the case of fast green, in 2 orders of magnitude from 11 to 1,200 picoseconds. So basically, we can conclude that most of the probes that we speculated that behave as molecular rotors and could be used in order to sensor physical properties of food were actually molecular rotors, and that any environmental restriction to intramolecular twisting results in an increase in fluorescence, quantum yield, intensity, and also lifetime. So it was the time to apply them to sample viscosity and see if they were able to report in time the characteristics in model food systems and also in in-time measurements. So we solved molecular rotors in solutions of mono and disaccharides. Very interesting sensitivity of these molecular rotors to the restriction caused by increasing viscosity, particularly for mono and disaccharide solution. So, interesting advantage in comparison to conventional rheometers, because most of shear rheometers cannot test viscosity of highly concentrated sugar solutions because the shear causes crystallization and they cannot be accurate measure. So basically, molecular rotors allow us for that application. To extend it even further, here what will happen with hydrocolloids. We use see that the sensitivity was still present. However, when we expanded to similar colloids such as carboxymethylcellulose, we could see that that sensitivity completely disappeared. Speculated that what we were observing was that, for example, the first samples that I showed you that were in glycerol, they basically create a tight packing around the probe. It happens with the saccharides. When we have hydrocolloid, the probe sits in a highly complex network where we basically sense the water that is being retained by the network itself and not the walls and the structure of the shell that is being formed. So, local sensitivity reports on basically water instead of the over environment. So there is a discrepancy between what happens at the local level and at the bulk level. In case the sucrose cause exactly the same effect at macro and bulk level, so that's why this molecular rotor was effective in order to characterize both the macro viscosity and the bulk viscosity. The thing that was interesting is that, you can see in the previous slide, some colloids it works perfectly, but for others it didn't. We were wondering how we test for the crowding. We use another probe that's called pyranine in order to assess differences at the local level. This particular probe, and of course this optical strategy, is on the characteristics of this particular compound, which exits a double bond. If it is protonated, that is more free water around the probe and exhibits a peak around 500. Unprotonated, the less water around the probe and peak shifts to 135. Typically, by calculating the ratio between those two peaks, we can figure out what is the crowding in terms of free water that the molecule exhibits. So, with two samples that solutions with equal viscosity, we measure the fluorescence intensity of Allura red in those, and also the fluorescence intensity for pyranine in both. Is that the light fluorescence intensity of Allura red goes down as the size of the used to modulate viscosity increases. So between the first and second peak for pyranine also decreases. What's interesting is that while the fluorescence intensity for cellulose is still relatively high, for CMC is extremely low. However, the environment around the probe seems to be the same. This suggests that particular components establish some kind of interaction with the molecular rotor, restricting their movement, and that's something that we can exploit if we identify those particular interactions in order to make these probes more sensitive to the physical environments of particular foods. Overall, we can say that several glass compounds exhibit molecular rotor behavior. Their sensitivity to changes in the physical properties is comparable to commonly reported molecular rotors. We have to bear in mind that this sensitivity is effective at the local state, and that course, that stems from they are molecular probes. In systems where the viscosity modulating agent affects the bulk and local environment to the same extent, they can constitute, therefore, absorbable viscosity. The interaction of the probe with the particular component will restrict its movement and will make it as a physical property probe properties that will know that interaction. In the case of one of our probes screen, we knew that a probe a dye that is commonly used in microscopy in order to characterize proteins. We know that it exists already an interaction with that can be exploited in inline viscosity measurements. And that's basically what we did. So, as food scientists and food engineers, one of the things that we strive the most is to report on an actual process. Of course, the main inline process that we want to report probably is digestion, either in vitro or in vivo. In digestion, we have physical modifications of the food itself, which can affect the digestibility and satiety. We want to figure out what is the bioaccessibility of the bioactive compounds that we have in that food. And also, in most of the cases, we are fortifying food. We know, so we try to figure what happens with those delivery systems as they go through the gut. So it's extremely interesting to have the ability to monitor those physical properties as the food transits through the GI tract to assess how those physical properties affect bioaccessibility and, if possible, satiety. So we use a molecular rotor, as I said before, in order to do so. We couple a digestion simulation system, the one which basically represents the GI tract, and it has a compartment for the stomach, the intestine, for the with a spectroscopic technique, and also assess bioaccessibility of the components that we were feeding to our system using an HPLC. One we have to take into consideration is that we are talking about a complex process, a complex food material. We didn't want it to test just a model system, but an actual food. It's of a complex process. Problems that we have is that there are some chemical factors that affect this particular system, as pH, the presence of enzymes, salts, biopolymers, and so forth. Also, in terms of the intestines, the presence of biosalts also contribute to the difficulty in order to perform this kind of measurements in line. And in terms of mechanical factors, you have periodic movements which are going to affect also the performance of an inline measurement. So in order to avoid false conclusions, we perform extensive preliminary testing for selection, testing the sensitivity of our molecular rotors to pH, IK strength, addition of enzymes, addition of bias. And all of them, a rotor, the one that is based on an edible probe, perform in a similar way as conventional molecular rotors, especially designed for a monitor viscosity model systems. Another thing that we wanted to obtain was appropriate differentiation. Remember that not only that the system is complex, but the food is complex as well. So we needed to have a good differentiation between signal from the itself and the autofluorescence of the food. So in the case of a particular unconventional molecular rotor, it was very difficult to obtain this differentiation, but when we went for the probe, we thought in our preliminary test that was going to perform better, it actually did. This particular probe, fast green, has an excitation wavelength of 500 nanometers, which is considered the far red. Most of the foods autofluorescence is elicited when we enter UV light. So, exposing this particular probe, a red light, provide us an appropriate differentiation of the probe emission in relation to fluorescence of the probe. Additionally, we correlated the presence of these of several solutions of the material that we wanted to be digested with the fluorescence intensity. We have a good correlation between the fluorescence intensity from the molecular rotor and viscosity itself. At that time, it was appropriate to test it in an actual system. So as a probe fast green, data collection was done using an excitation within the far red in order to produce appropriate differentiation of probe and the autofluorescence of the sample. And we perform all the measurements on a Carry Eclipse equipped with a fiber optic, which allow us to make measurements in each of the compartments of the simulator of the digested system. We were lucky enough that all these compartments are made of glass. We didn't have any problem in terms of having to verify that one. In terms of the results, we follow digestion through time and can see how the fluorescence intensity diminishes as the digestion of the sample proceeds. What's interesting is that we compare two grain products, pre-gelatinized one and coarsely cut processed grain. In the case of the pre-gelatinized grain product, we would observe that the viscosity of the solution that we feed to the team diminished as time went by, while in the case of the processed grain, we observe an increase in viscosity at almost 2 hours of digestion, which of course might have some implications in terms of the satiety of a particular sample, and made a case in terms of consuming highly processed versus processed grains. Not only the changes in viscosity were interesting, but also they relate with changes in bioaccessibility of sugars. These data were obtained using HPLC, and in this case, the bioaccessible sugar for pre-gelatinized product was much higher than that obtained for the coarsely processed grains. So basically, in terms of using molecular rotors as probes for physical states during digestion, we can say that with the appropriate selection of a probe, they result in sensitivity. These data are extremely easy to collect, extremely easy to analyze. Actually, depending on the probe selected, they're not only applicable to physical states, but can be extended to other properties. In terms of during digestion, this is not only limited to the equipment that we use, the Team 1 simulation model, but it can also be expanded, for example, to a static simulation system. They are more common in most labs. For example, a dynamic system, probably even with the addition of the microbiome. And of course, the ability to measure the physical state of a food that's been digested might have interesting risks in terms of the food within that produces into satiety, which might bat the growing obesity epidemic. This was not the only use that we found for molecular rotors. Wanted to test was the viscosity of a liquid soil in a fat crystal network. So basically, in case mixture of fats and oils, the idea is to understand fat blooming in chocolate confections and coatings. So we use our blanks without the addition of the probe, and the same seed with the addition of citrus red, which is an edible synthetic dye approved for use on coating of oranges, especially in United States. We measure the emission for both systems, and we correlate with the fractal dimension of the fat networks and with solid fat content. So you can see that as the solid fat content increased, also did the normalized fluorescence intensity. But what's more interesting, when we calculated the normalized intensity with a proxy for the microviscosity of the oil entrapped in a crystal network, it typically shows two regimes. Once below a fractal dimension of 1.89, which means that the confinement did not alter the microviscosity of the oil. Above 0.989, in each case, the viscosity of the bulk oil was representative of the oil in the system, and the microviscosity was provide a better correlation. It's extremely important in order to figure out what happens in actual systems, which most of them has a fractal dimension above 1.89. And the press-fade technique that allows us to figure out what happens with microviscosity during entrapment would be a plus in order to develop materials with chance characteristics and stability, especially in terms of fat blooms. Other applications and uses of molecular rotors non-reported non-perfect comes to the use of phase transitions, of molecular rotors in phase transitions, for example, volatility of gases, flow polymerization, and also of sensors of flow, which might have interesting implications in terms of fluidics, especially now that many of us are working in lab-in-a-chip devices. In this case, you can observe that as the flow rate decreases, also the emission of a particular molecular rotor. The mechanisms of this particular performance are still being investigated. Probably one of the most interesting applications currently used for molecular rotors, and that hasn't been used or expanded in foods, is their application in fluorescence lifetime imaging. So this one tends to be representation of a microscopy. which equips the photocounting unit that allows to measure for lifetime. We will have a sample that has different domains. Let's either, for example, a cell or a system such as, for example, a multiple emulsion that will have the interfaces and lead these liquid continuous phases. So the sample is excited with the laser and separated or divided using a grid. The time selected for each of the in that grid allow us to map the differential sensitivity and viscosity of particular sections of this image. So, molecular rotor that is associated to interfaces, of course, that's a much more restricted environment because it's attached to that interface, which also responds or in intensity, while the body is flowing inside the liquid medium, going to be way and there are not going to be restrictions in terms of attaining the DICT state. All results in lower viscosity. By mapping each of the particular parts of this image, we will be able to establish different lifetimes to the performance of that molecular rotor in different environments with different viscosities. So that now a complete map of that particular image can be created that shows the regions with higher viscosity and lower viscosity, for example, inside the cell or in the internal continuous phase of a multiple emulsion. These techniques has not been applied in food yet. They might be promising in order to figure out what happens at the interface, understand fluidity and emotions and dynamics in a delivery system. The only applications that optical spectroscopy can found in foods. For example, in case of following processes, the diffusion of a liquid oil in fat mixtures is something that can be attained using FRAP, which is fluorescence recovery after photobleaching. And the technique is quite simple. You have a particular sample which is stained with light. To a high intensity light, only a region of that particular sample is being affected, the one that has received the direct incidence of that high intensity light, and become decolored, or actually the emission is basically neutralized because of the radiation of the light in that region. And now you keep on observing the sample under the microscope a long time. It allows you to figure out what is the diffusion of the surrounding dye in the region that has been affected by the photobleaching. Here you have an actual sample. In this case, it's been deemed the zone of interest, which is basically the use of an intensive light source. After it has been reached, the essence is being observed in this region. And once you wait for some time and allow the diffusion of the surrounding probe into the depleted zone, you can fluorescence recovery the region of interest. Use Corradini and Rousseau in order to calculate the diffusivity in mixed systems. In this case, you can see that fluoresces, which is the lightly color, and that one is the one that doesn't emit any fluorescence. Time goes by, you get an increase in fluorescence intensity due to diffusion, and in the second figure, they were able to translate data into diffusivity and responded to solid fat content. In a similar manner, we have developed easy measurements in which we don't use FRAP, but we use an optical measurement in order to follow function of red fat mixture on time and do measurements in real time as the diffusion happens. Basically, we use this device that is called transfusion cell, which has a donor chamber. We place a sample in between the donor chamber and the receptor chamber. It has, of course, its water jacket in order to perform the study at the constant temperature, and it has a sampling port. Sampling port, we use it in order to include a fiber optic that was to measure the sense of the diffuse probe with and corroborated that was diffusing at the same rate as the compound of interest, a liquid oil, and we measure fluorescence a long time. The spectrophotometer. The results of this experiment allow us to see how we have an increase in fluorescence as time went by, using, of course, a simplified version of physics law. We were able to the diffusivity at each time. This allows us to correlate diffusivity with solid fraction and also with fractal dimension. And that has important implications in terms of development of appropriate fat mixtures in order to reduce fat blooming and fat migration, which is normally not desirable in many chocolate candies. Let's discuss a little bit potential use of optical measurements in order to monitor formation and disassembly in delivery systems. Our work in this area basically covers the formation and degradation of protein-based delivery systems obtained by clear anti-solvent precipitation. Advantages of using molecular techniques is that we can test delivery systems with minimal alteration of the structure. Because, first of all, we don't have to use labels with no dilutions. For example, in most, every time that test particles of a delivery system, of course, that system has to be diluted, which is not something that most of the time you can do. And of course, you are not applying any kind of mechanical stress. So basically, you are not deforming your sample while being tested. And I'm going to introduce another concept that is called Foster resonance energy transfer, because that's the one that we use in order to what was happening during formation of protein delivery system. In case of Foster resonance energy transfer, or FRET, you have a loop that basically excites when it is exposed to a particular light source, and an acceptor, which is another probe that can also pass when exposed to a higher wavelength. The spectra of the emission of the donor does not overlap with the actual excitation you don't fret, but if they overlap and the samples are close enough, you will have emission from the acceptor instead of having emission from the donor. So if you have no fret, one of the things that you are going to see, you're going to excite your sample. If the donor is going to be excited, I'm going to see one peak that corresponds to the itself. This happens because the donor emission and the acceptor excitation are not overlapping, or they are placed at a long distance from each other. We foster distance, and hence the name of this technique. So if they are far away, you see the emission from the donor. However, when they come close together, they don't transfer the energy to acceptor. The acceptor excites and emits. Instead of seeing the spectra, the spectrum corresponding to the donor, you see the spectrum corresponding to the that was to out if the donor and the acceptor are really close together or far apart. This is particularly important in proteins because it allows us, for example, to assess interactions on formational changes. If I have two proteins, one being the donor and another being the acceptor, when they are coming together and interactive, you are going to see fret and emission from the donor if they are close together. This happens when you are having conformational change. You have an acceptor far from each other in the same, for example, protein. Once you take characteristics of environment, let's say, for example, checking the pH or the acidity of the solution, and come close together, now fret can occur. Now you can infer that those of the molecule are coming together. Now you is properly sized to infer what's happening in terms of changes at the molecular level. In base molecular systems, now it's quite popular the use of liquid anti-solvent precipitation in order to obtain this food-grade-based delivery system. One of the things that happens is that the measurement of the formation mechanisms is a little bit cumbersome to follow because the formation is too fast and the system shifts during testing. In case of anti-solvent precipitation, a protein solution is included into a solution that is not considered an anti-solvent because a particular protein that is dissolved in this system collapses or forms or changes conformation once it's in a solution doesn't ask for its proper hydration. So what happens is that because of the change in solvent, it forms small particles that we use as delivery systems. Proteins tryptophan and tyrosine in their composition, which are both same probes with the aromatic groups that they have. and the cluster distance between, in order to have that faster energy transfer, is 10 to 19 angstroms. Usually, tyrosine transfer into tryptophan. In this case, we evaluated two systems, one by gliadin, other form with whey protein. Gliadin is not soluble in water, but is soluble in ethanol, so the anti-solvent for gliadin was water. We dissolved it, and we kept on adding water, and we could see that the intensity of tyrosine was being diminished upon formation. We would speculate and are doing additional studies in order to figure out if we are actually point. However, the preliminary data suggests that tryptophan is coming closer to tryptophan, having fret this energy transfer, which reduces the intensity that we were observing at the initial part of the test. And as the system is being formed and collapsing into itself, the tyrosine comes closer to the tryptophan, and the emission from tyrosine is being transferred to that acceptor. This happens because gliadin is a really stretchy and linear protein in comparison to, for example, a protein, which is our next example. So for this particular case, this internal probe tyrosine can be considered a good intrinsic probe to monitor the formation of this system. Of globular proteins such as whey protein, the thread between tryptophan and tyrosine happens from the beginning. But we could use the intensity, the emission intensity of the tryptophan itself in order to follow formation. Not only the intensity was sensitive to formation, but also the location. We were observing as the environment around the probe changed and the whey protein aggregates upon itself. We could see a shift to a higher location, a shift to the red. So in this particular case, this intrinsic probe also a good source to monitor the formation in this particular system. At this point, we are doing further studies in order to see the feasibility of independently verifying this finding. Shelf life monitoring use spectroscopy in order to handle evaluate variability due to handle discrepancies between actual and nominal shelf life. There is a huge movement nowadays that suggests that we are throwing away perfectly good food, and probably they are right, because we are basically evaluating shelf life based on predictive models and with large safety margins, and not making measurements in terms of in real time. If we could identify fluorophores susceptible, for example, to thermal abuse or to changes related to safety concerns, we could reduce waste without increasing chemical or microbiological risks by following what happens in real time in our food. So, but this will imply of following changes in essence fingerprints instead of changes of a single fluorophore. It's a little bit more complex, but it has been effectively done using hyperspectral analysis, either based on fluorescent measurements or absorbance and reflectance measurements. And as I mentioned before, for example, in this one, see a proliferation of bacteria of a food sample as a function of time. Now the fingerprints, the fluorescent fingerprints of this particular chain a long time, where and this has been correlated with microbial counts. So we can see that at the beginning it has about 4 log cycles, and it went up to 9.7 according to the authors. Both Zoom and Badramidis have in the last year reviews on hyperspectral analysis. So if you're interested in these techniques, I suggest that you consult this literature. So let's sum up and let's summarize the impact of these techniques in science and engineering. Well, first of all, they provide a non-invasive, non-disruptive, highly sensitive analytical methodology. They allow us to evaluate materials, the systems, and actual foods as they are. They also allow us to relate complex spectral parameters of optical emission to specific molecular properties and even to mechanisms in food and food materials. And also, they allow us to monitor quality, stability, availability, and safety of the food supply at various stages of the food production and distribution or the point of sale, if properly articulated. And on the use of luminescence spectroscopy in food and food engineering can provide a good source of analytical tools and new strategies in order to evaluate our food supply in a minimal invasive way, and also with relatively low cost. Well, I would like to thank Berti and Vinicius Armen Moreno for the invitation to give this seminar. So Dr. Gustavo Barbosa Cánovas for his promotion of this seminar. As well as I would like to express my appreciation for my collaborators, Dr. Michael Roach from University of Wales, Bogomil Lent, and Siri Sinski from UPenn and UNT, Richard Ludesher, Dipti Usalvi, and Mukwawe from Rutgers University, Iris Joy from UK Leuven. And I will be glad to answer some questions.
Well, thank you, Dr. Corradini, for your presentation. Attendees are able to unmute their microphones to ask Dr. Corradini any question. Dr. Yes. Yeah, we hear you now.
No, thank you very much for this very good presentation. I don't have a specific question. I have some about the use of these methodologies, these technologies, to improve the knowledge in very different topics, very different areas of food science, food technology, and food engineering. What is your opinion of the future of application of these methodologies to improve the knowledge in some specific fields, some specific areas?
Well, I think that mostly. I think that in order to monitor the biophysics digestion, these technologies are going to be extremely useful. And I think that they will not be only able to allow us to monitor what happens in terms of the physical state of food, but also, for example, there are many of the bioactive compounds that we want to add to foods, and that we can monitor their stability as digestion proceeds without having to intervene or stop the process. Also, in terms of medical applications, I think that a lot of people are extremely terrified about ingesting foods that are not edible to their own bodies and having be monitored by strenuous techniques. So now they are developing the spectrofluorimeters that are extremely small, and they can be taken as pills. And basically, one could apply this in biomedical applications. So basically, in order to figure out what happens in the digestive tract, it would be a really interesting application. However, there is an important caveat. As long as we don't devote time in order to figure out the sensitivity of this particular particular properties, we will not be able to pinpoint these correlations. So we have to invest time in preliminary studies in order to address those things.
Thank you very much.
Thank you.
Dr. Kenny? In case of among all the tested probes and these spectroscopic techniques, which one things that can be implemented in commercial food applications in the shortest time span? Which one do you foresee?
Well, I think that the direct measurements, I think that the ones that are based on autofluorescence are currently being applied. And so, really, the problem is not making the data acquisition, but the data interpretation. A lot of people are working with chemometrics in order to understand the contributions of each particular probe and the overall fingerprint of a sample. And I think that in terms of research and development, the application, for example, of FRET should be relatively easy and advanced that could be implemented, for example, in terms of evaluating what's happening in delivery systems. And FLIM is also going to be extremely used in food research if we figure out which are the adequate probes and with increased resolution of fluorescence microscopy and photon counting devices, which is coming. I think that within probably 1 year we will be able to use that for research. So I think that all the methodologies that I presented have extremely possibilities of being commercialized to some level or the other, either for just research or for actual applications. In terms of the sensors, I think that, for example, in the case of riboflavin is thermolabile. So implementing it as a temperature abuse probe might be something extremely feasible in no time.
Okay, thank you, Dr. Corradini. I have another question regarding the last application, or maybe curiosity, but the type of microorganisms, have images profiles been tested for a specific microorganism, total counts, or?
Yeah, they have been done, particularly for Escherichia coli, in surfaces. They've been done also in terms of meat with some other microorganisms. And none of them, all of them are label-free, so it depends on the autofluorescence of the sample.
Okay.
So that has been a relatively large amount of advance in that area. Particularly Valdramidis has done a lot in terms of microbial, if you are interested.
Yeah, I'm interested.
It's fun.
Anyone in the audience who may want to ask another question for Dr. Corradini? Well, I would just like to thank you once again for accepting our invitation for this excellent presentation, and we'll wish all of you a nice day, Dr. Corradini.
Thanks so much for the invitation. Bye.